So, cell transport. Now, obviously things need to move across the cell membrane because you need to bring particles in, so you need to be able to bring sugars and nutrients in, and particles that are functional in terms of building the cell or functional in terms of metabolic processes. And at the same time, you also need to get rid of particles all the time because, of course, your cell going through respiration is going to be producing even, and as an example, carbon dioxide, and you're going to need to get rid of that carbon dioxide because if it builds up too long, it can become toxic within the cell. So things need to move across the cell membrane, and this is referred to as transport. Now, there's a couple of transport mechanisms. There's passive transport. Now, passive by definition means that it doesn't require a ton of energy, it just happens. Now, there's two types of passive transport. There's diffusion and facilitated diffusion. And then the extra is osmosis, three types. There is diffusion, facilitated diffusion, and osmosis. And we're going to break those down. So this is just a general response, and then we're going to break them down into their parts. Then we have what's called active or energy requiring transport. So there's active transport. Passive doesn't require energy, and active requires some kind of energy investment in order to move things across the membrane. Active transport has these things called ion pumps, and we'll talk about ion pumps. It has something called cytosis, and we'll talk about cytosis, endocytosis, exocytosis. That's just where the structure of the membrane is involved in moving things into the cell. And ion pumps just means these proteins, these integral proteins on the outside, are actually pumping particles across the membrane against the density. So there might be a higher density inside the membrane, and they're trying to increase the number of particles even though it's denser inside the membrane than it is outside. And that means that it can't just use diffusion. Okay, so this is an example of our membrane. We have the plasma membrane just here, so that's the plasma membrane in the middle here. We have the cell cytoplasm, the blue bit on this side, so the goo inside the cell. And then on the outside, we have the outside environment. So we have different particles on the outside, and we have different particles on the inside. And we need to manage the density of those particles. So a cell needs to manage the density of those particles. So the cell membrane is partly permeable. We've talked about the fact that it's partly permeable. So for instance, water can just naturally move across the cell membrane. It doesn't need tubes, it doesn't need pumps. It just naturally moves, and it moves from an area of high density to an area of low density. But however, not all particles can move across, so it's semi-permeable. So some particles just can't move across it, and they need to be moved across it. Okay, so that's partly permeable for certain molecules. Okay, we talked about channel and carrier proteins and the fact that they're involved in transport of some molecules and ions across the membrane, and we talked about that for the last sub-lecture. And then we can have things like these large particles, like proteins, like very big particles, and those big particles require transport through what's called cytosis, and we're going to talk about that later in this sub-lecture. So the first concept, and one of the most important concepts in all of biology, is this concept of diffusion. Diffusion is the movement of particles from an area of high density to an area of low density. So particles naturally move from a place of high density to an area of low density in liquids and in gases. It doesn't require energy to do so. Diffusion describes the movement of molecules from regions of high density to regions of high concentration to regions of low concentration across what's called a concentration gradient. So the gradient is from high concentration across to low concentration. In biological systems, diffusion often occurs across partially permeable membranes. So you can have a cell membrane, for instance, as a partially permeable membrane. So the example that I give my students in class because it sticks in their brains. It's not a particularly polite example, but it's an interesting one, is the idea that if you, for instance, were to fart in the corner of the room, then eventually all those particles from that fart would spread out into the room, and unfortunately your neighbours would eventually smell it because those particles are spreading out. So they diffuse. They go from the area of high density and they slowly move outwards to area of low density, to the effect that eventually the particles are so far apart and they're at so low densities as they spread out that you can no longer smell the fart, thank goodness. Okay, so diffusion across a membrane. Okay, so in any type of diffusion, each type of diffusing molecule, the gas, the solvent, or solute, moves along its own concentration gradient. So it's independent of the other molecules, it moves on its own concentration gradient. So you can see over here, the red particles are at higher concentrations on this side of the membrane, so they're going to slowly diffuse across the membrane so that you'll have even number of red particles here as there are over there. And the blue particles are going to move from this side over to this side, so that eventually you will have an even number of blue particles on both sides. So that's how diffusion functions. An equilibrium is reached when the net concentration of molecules on each side of the membrane are equal. At this point, we have what's called the net movement stops. So what that's talking about is this kind of dynamic equilibrium, is this idea that particles will still be moving across the membrane, but the rate of movement will mean that it'll be even concentrations of those particles inside and outside. Two-way diffusion across a partially permeable membrane is common in biological systems. For example, at the lung surface, carbon dioxide diffuses out of the blood across the membranes of the capillaries in the lungs and the alveoli, while oxygen diffuses in the opposite direction. So all that's saying is that when you breathe in, the density of oxygen in your indrawn breath is then higher than the density of oxygen in the blood that's coming from your veins, because they've dumped all that oxygen in your cells. And so consequently, the oxygen in the indrawn breath is diffused across into the capillaries, the little kind of veins in your alveoli, the sacs in your lungs. And so naturally, the oxygen actually just moves across into your red blood cells. At the same time, carbon dioxide, which is now at a high density in your veins because of the fact that it's been added from all your cells at the end of respiration, actually naturally moves the other way and moves into the spaces in the alveoli, and then you breathe it out at a higher density than it was when you breathed in. Okay, now there is two types of diffusion which I want you to know. There is just simple diffusion, which is the one on the other side there, and this is what's referred to as non-polar molecules. It's just molecules that don't have a really defined charge, right? So they're not charged differently across the molecule, and they're quite simple molecules. So an example is oxygen and carbon dioxide and water, and those things can just naturally move across the membrane without being helped much. They just naturally move across. And then we have what's called facilitated diffusion. So these are larger polar substances, i.e. ions, and macromolecules is large, and they can't just naturally move straight across the phospholipid bilayer the way that carbon dioxide and oxygen can. So what they need to do is they need to take advantage of these carrier proteins and channel proteins. Now, they still move just through diffusion, so there's no need to use energy to move them across. They naturally move across, but they need these tubes, these channels to move through, because they can't just push through the phospholipid bilayer. So all I need you to know is that you have simple diffusion for these simple small molecules that are non-polar, and we have facilitated diffusion, just facilitated by the structure of the protein. So facilitated means helped. Well, it's just the structure of the protein that facilitates them. It's not active, it doesn't cost energy for your cell, it just naturally moves through that structure. Okay, so we've hit diffusion, and this is just showing you that the hydrophobic molecules and small uncharged molecules, so you can see there the carbon dioxide, the nitrogen gas, the oxygen gas, and the water and glycerol, they naturally move through using simple diffusion because they can just push between the phospholipid bilayer. Then you have facilitated a diffusion for the ions. So these are ions down here. When you did chemistry, you talked about ions. So they're charged particles. They're quite charged particles. And so as a consequence, they can't push through the phospholipid bilayer. So they bounce off. And they therefore need facilitated diffusion. So what they need is those protein channels. So the integral proteins, which we were talking about in the last sub-lecture, and those integral proteins allow them to move and push through. They still don't need energy though. Okay, so that's just a diagram showing what I've just talked about. And by the way, all of these, you could access diagrams just like this online, just to simplify your note taking, etc. And the same could be true of the previous sub-lecture, and you're talking about different particles in the cell membrane. You could make a really nice kind of graphic representation of that really well, and I would recommend that you do that because it's, turning it into a table is a great idea in terms of your own revision. Okay, the next concept for passive movement across a membrane is osmosis, another really important and interesting biological process. Osmosis is a passive movement of water molecules, so specific to water molecules, and it requires no energy because it's passive. By definition, it's passive, so it requires no energy. And water molecules move across a partially permeable membrane from a region of lower solute concentration to a region of higher solute concentration. Okay, now what that means is this. An example we're going to use is salts. So if you are a cell in a really salty environment, so the Dead Sea, for instance, a cell in the Dead Sea, then the density of salt, the concentration of salt in solution outside the cell, if we put the cell in the Dead Sea, the concentration of salt in solution around the cell would be greater than the concentration of salt inside the cell. Now what that means is that because of osmosis, the water inside the cell will naturally move outwards to even up the ratio. Now when it does that, it'll suck water out of the cell. It'll suck water out of the cell. Now you can test this really easily with a microscope and a plant cell, like Eulodia plant cell. You just take a plant cell, take some plant cells, you look at it under a microscope, and you introduce a saline solution. And what will happen is a cell membrane will suck away from the walls, and because all the water inside it will actually push outside. And the opposite is always also true, and that's what we're going to quickly have a look at. So this is a really important concept in biology, and it affects organisms all the time. And the example that I've given you there is the reason, apparently, now look, I assume this is accurate, but apparently the reason that bull sharks are called bull sharks is they come up into freshwater systems, and when they do, because of osmosis, their cells actually absorb their body absorbs a whole ton of fresh water, and it naturally moves inside, and they get all bloated and fat. And so the consequence is they're called bull sharks because they look really bully and fat when they're in freshwater systems, which is really cool. Okay, so we've talked about this really quickly before. All this is saying is that plasmolysis is the tendency for the cell membrane of plant cells to suck away from the cell walls and leave the cell walls when they're in what's called a hypertonic salt solution. Now we'll describe hypertonic and hypotonic, but hypertonic is just saying that if the solution around the outside has more salt in it, then the solution inside the cell, in the cytoplasm, the water is actually going to move out of the cell membrane into the surrounding environment, and as a consequence, it's going to suck the cell membrane inwards, and you're going to see this kind of spikiness in the cell membrane. Now, usually cell membrane would push up against the cell walls, but it actually will suck inwards. Now, the opposite is also true. So if the solution around the outside is hypotonic rather than hypertonic, that means that there is less salt in the solution outside than there is inside the cell. And what's going to happen is the water is going to move inwards to even it out. And actually the cell will expand, and cells can pop that way. They can actually pop. And this is a bit more of a descriptor of that process. So there are three options. Animal cell environments. Animal cells lack those cell walls that plants have. And as a consequence, they're really affected by the surrounding environment and the solution that they're in. Without a rigid cell wall, animal cells may change shape depending on the conditions of the cellular environment. And we're going to use a red blood cell because it is a really cool example that you can literally see under a microscope. In an isotonic solution, so there's three solution types. In an isotonic solution, the cell, such as a red blood cell, maintains a normal shape as there's no net difference, like there's no net movement of water into and out of the cell. Like it's moving across it, but it isn't all moving outwards or all moving inwards at a higher rate, with the consequence that the cell stays its natural shape. So they're kind of donut-shaped red blood cells. Cool, so that's an isotonic solution. So that really means that the concentration of outside to inside is similar. So that's isotonic. Now, it keeps its ordinary shape. However, if the surrounding solution is what's called hypertonic, which means that the water potential of the extracellular environment, the outside environment, is less negative, so it has a lower solute concentration, okay? Lower solute concentration. Think of the example of salt. So there's less salt in the outside environment than there is inside. Now, solute doesn't always have to be salt, but we'll use that example because it's a nice, easy one and a common one. If there's less salt outside, then water is actually going to move into. So less salt outside is hypertonic, or less solute concentration, lower solute concentration outside, then water is actually going to move into the cell. So in a hypertonic environment, cells swell because of the net gain of water across the plasma membrane. This red blood cell, you can see it's big, fat, and juicy, will eventually burst. It'll literally pop. It'll lyse, so it'll pop, because this membrane cannot withstand the pressure of all this extra water. In a hypertonic environment, water will enter the cell because a cell cytoplasm has a higher solute concentration, and we're using the example of salt, but solute concentration of all these particles inside than there are outside, so water is going to tend to go into. In an animal cell with no cell wall, this will eventually cause the cell to lyse, to go through lysis. The cell, i.e., will burst. In cells surrounded by a cell wall, e.g. plant cells, the cell wall provides rigidity, and the wall pressure will counteract the push of the water, but you and I don't have cell walls. And this is the opposite, and this is really cool as well. So rather than a hypotonic solution, if they are in a hypertonic solution, which means that there's a higher solute concentration, using the example of salt, right? If there's more salt outside the cell walls, or higher solute concentration, then what will happen is the water inside the cell will start to move out of the cell. And so the cell membrane will start to shrink. And you can see, because it's an animal cell, it has the cytoskeleton, and the cytoskeleton is making it all spiky, but the cell membrane is pulling inwards, which is really cool. In a hypertonic environment, the cell will lose water because the cellular environment has a higher solute concentration. The term for this process in animal cells is crenation. In plant cells, the process is termed plasmolysis, which we looked at before. A cell can lose its ability to function if it becomes too severely dehydrated, and you can literally do this to red blood cells, and they just suck inwards. They're amazing. They look all spiky, this is an electron micrograph. It's a nice close shot of that, which is really cool. Okay, so we've talked about passive transport. Let's quickly talk about active transport. Now, diffusion alone cannot supply the cell's requirements for molecules, as some are required by the cell in higher concentrations, which we've talked about before, then occur outside the cell. So sometimes we just need more or higher concentration inside the cell than is naturally occurring outside the cell. So we actually need to actively pull molecules into the cell. Active transport is the movement of substances across the membrane using energy. So if you can know that passive transport doesn't use energy, but active transport requires energy in the form of ATP, which we'll talk about later. Now, just know that it requires energy, and that energy, the form of that is ATP. We will get into that in future lectures, but it requires energy to move things across. Active transport can move substances against their concentration gradient. So diffusion or passive transport must be with the concentration gradient from high concentration to low concentration, but active transport can take them from low concentration towards high concentration and increase the concentration, but it requires energy consumption. Okay, so active transport. The work of active transport is performed by protein pumps in the membrane. So these are those integral proteins, a different integral protein that literally pumps things into the membrane. So the particle smacks into the outside randomly. It is then captured and then pulled inside. But in the process, you can see it uses ATP and turns it into ADP, and that is consuming energy. These proteins pumps harness the energy of ATP, adenosine triphosphate, to pump molecules from a low to a high concentration. And that's just another example of the molecule. The protein changes its shape in such a way as to move the bound molecule across the membrane, so it literally kind of captures it and pulls it inside. So cells can control what is absorbed and what is expelled. So if you can just know that it requires ATP and it can kind of capture a molecule and push it against a concentration gradient, you've had a win. Now, the next one for active transport is just an ion pump. Now, all an ion pump does is this. Ion, referring to the fact, and you've talked about ions in the chemistry part of the course, ions are these charged particles, like that's the sodium +1 there, the Na +1, the sodium. And what it does is the sodium pump is a protein, so it's an integral protein, the same as the others. It's an integral protein in the membrane that exchanges sodium ions for potassium ions. So the sodium is on the inside at a higher concentration. As it moves outwards, it then grabs a potassium ion and pulls one inwards. ATP is used as the energy source for the exchange, and the unequal balance of Na plus and K plus across the membrane creates large gradients in ion concentration, which can be used to drive other active transport mechanisms. All I need you to know is that you can have this active transport using what's called a sodium pump, and that it's a charged sodium. That's all I need you to know. Okay, now last but not least for active transport, this is cytosis. Now it's relatively large, but we'll try to knock off really easy. Cytosis is to do with the use of membranes for moving larger particles. So these phospholipid membranes are semi-fluid, and they can actually move particles by making like a bubble, and that is cytosis. It's a form of active transport involving the formation of membrane-bound vesicles or vacuoles. Regions of the plasma membrane become enfolded, invaginate, so fold inwards, or evaginate, which means to fold outwards, to transport substances across the membrane. Cells are able to do this because of the mobile nature of the membrane. It's just saying because they're fluid, like we talked about last sub-lecture, they're actually able to pull inwards with the membrane and particles come into that kind of bubble. And then they can pinch away from the membrane and move into the cell. Or the particles can be in a vesicle. The vesicle can hit the membrane and push the particles outwards into the surrounding environment. Now, they can carry out cytosis in the form of endocytosis and exocytosis. All about words. Biology is all about words. I've said it from the beginning. Endo means inwards, inside, means to pull things inwards. Exo means to push things outwards. Exo means outside. Consequently, endo, inwards, exo, outwards. X is outside. Now, the way that I would try to remember that is to remember that a insect has an exoskeleton. So as it's outside skeleton. And you guys are endo. Your skeleton is endo. It's inside. Cool. Okay, so endocytosis, think bringing things inside, and that just describes bringing substances from the outside the cell to the inside the cell using a membrane-bound vesicle. So that up there is a solid particle, a big chunky particle. So that wouldn't naturally even move through one of those proteins. So these are big chunky particles. They might be a piece of a cell. They might in fact be a cell. So your white blood cells use this to surround bacteria, and then they form a vesicle around it and bring it inwards, and then they push a whole ton of enzymes into the vesicle, and the enzymes then break down that cell and destroy it. And then they might even use parts of that cell. So you can see that big solid chunky thing, it's being surrounded, invagulated, and it surrounds the particle, and then it makes a membrane and a vesicle around it, and then it pulls it inwards to potentially be broken down. There are 3 forms of endocytosis, but don't panic about that. If you can identify that there's this concept of endocytosis, and endo means to bring things in, and it has a vesicle, a membrane that surrounds it, then you've had a win. But there are 3 forms. There's phagocytosis, which is the engulfing of a big solid particle. There's pinocytosis, which is the engulfment of liquid particles. And there's receptor-mediated engulfment, which just means that there's like a receptor on the outside that triggers engulfment. So this is endocytosis. You can see the particles are on the outside, and then the cell membrane invagulates, it pinches off. You can see it pinching here. And then you have this vesicle on the inside that then the cell can interact with, potentially put enzymes in to break down. Okay, then we have exocytosis. So think outside movement. Exocytosis, this is when we're getting rid of substances that could be waste products, there could be a broken down bacteria that your white blood cells have destroyed, and they release substances from the inside to the outside. So this is a transport vesicle, it's smacking into the cell membrane, and then what's going to happen is the vesicle, the membrane of the vesicle will integrate into the cell membrane and dump those particles into the outside. So if you can identify the fact that you have exocytosis and the exo is pushing it outwards, endo is pulling it inwards, and this is what it looks like. It's literally the opposite of endocytosis. You've got a vesicle down here. The vesicle has these waste particles or whatever they are, for instance, these could be enzymes, and they could be about to be transferred and put out into the surrounding environment. So they could be enzymes, and they could be useful in your body, in some way in your body for communication. And so what's happening is they're coming to the cell surface, to the cell membrane, and then they're dumping those particles out into the surrounding environment. Okay, so endocytosis we've got on the far side there. Part of the plasma membrane pulls inwards, and a droplet of liquid becomes enclosed when the vesicle is pinched off, and these vesicles can then move through the cytoplasm carrying their content. So they might have, like I said, they might have some kind of nutrient particle, etc. And then you can see there, exocytosis, the vesicle fuses with the plasma membrane and dumps the contents in the outside. But you can see that this diagram is a great one because it's talking about the fact that you might, for instance, have had proteins that are developed in the rough endoplasmic reticulum, and then those proteins have moved into a vesicle that's moved to the Golgi apparatus. The Golgi apparatus has put it into a transport vesicle, and that vesicle buds off, so this might have enzymes in it, it then buds off the Golgi apparatus and then connects and exports its enzymes out into the surrounding environment. So there's a couple of uses for that exocytosis process. It could be dumping waste, it could be dumping other particles that are of no use or toxic, etc., or it could be dumping enzymes for communication. And that's just a diagram that I found that I think is quite a good one for showing you transport broken down and the information for that transport. So we've been through that, but I think that's a really nice diagram. And you could look for a bunch of others online. There's literally tons of them, and cut and paste one of those or rewrite it or whatever you want to do. Okay, ladies and gents, that's all done with that one.